Welcome back, friends. So this is second part for chitin degrading bacteria. Okay, so this is a practical related video, and there is first part uploaded on my channel which describes everything about chitinase enzyme. So as we are here dealing with isolation of chitin degrading bacteria, the degrading here is actually meant by the enzyme, that is chitinase. Okay, so first, to study that particular chitinase enzyme, we need to isolate chitin degrading bacteria, and that is the main motto of your practical. Okay, so how to proceed for such kind of um, isolation, okay, where you want to see whether a particular biopolymer is being degraded by bacteria or not. So here we are going to study for chitin degrading bacteria. Okay, so this is part 2, and in this part at the end I will be discussing some random Viva questions just to give you an idea on what all questions can come on this particular topic. There is one more video uploaded on my channel which describes about chitin, chitosan and cellulose. Okay, so all these biopolymers, they are different. Okay, so what are the differences and where you find all these biopolymers and all those facts are all discussed there. So if you have this topic for your practical exams, I will say that please go through all these 3 videos including this part and be prepared for your Viva questions. Okay, because you will not just get questions on your practicals, you need to know the background as well. You need to know the facts related to the chitin, chitosan and cellulose. Okay, so let's start with today's video. So first part is isolation. So source of bacterial isolation. So this means the sample which you will be using for your isolation. So the sample can be soil, okay, then aquatic environment, then the gastrointestinal tract of a range of animal species including invertebrates, fish, reptiles, birds and mammals. Okay, so you can use any of these samples. Okay, so for example, to be very easy for you to go or to at least start for such kind of practicals, I will say that go to your nearby fish market and see where the waste product from the fish market is being dumped. So you can get a soil sample from that particular area, or you can simply get a shell of a shrimp, or I can see fish uh, shells, okay, or the gills part which is thrown out. You can just get those parts and you can proceed in your lab for isolation. But if you are not having any of these samples available, you can go for soil. So nearby aquatic environment, say you have a river nearby, then you can get a soil from the riverbed. Okay, so for example, we will now proceed with soil. So if you have a soil sample, you add that in 10 ml of distilled water, mix well and perform serial dilutions. So as we know, soil is uh, or it has a variety of microbes growing together. So you need to perform serial dilution. Okay, you cannot just go with a loop full of your suspension and you proceed for streaking. That will not cover all the bacteria which are present in your soil sample. Okay, so you perform serial dilution and you try to plate out at least last 3 or 4 dilutions. So that will help you cover all the microbes which are present in your sample. Okay, so you sprayed out or pour plate. So this depends on you which technique you want to proceed or you want to choose or use, that depends on you. So after doing plating, you incubate your plates, okay, at 37 degrees Celsius for at least 48 hours. Okay, why 48 hours? Because here we are actually observing the enzyme secretion, okay, the action of enzyme, whether the bacteria is able to produce chitin degrading enzyme or not. Okay, so for that at least I will say you should wait for 48 hours and then you observe your results. Correct. So this is the basic flow chart for how to proceed for chitin degrading bacterial isolation. Okay, so here I may keep on saying bacterial, but it does not mean that you just have bacteria which secrete chitinase enzymes. So there are even fungi present and known which are able to produce chitinase enzymes. So even you may get a fungal isolate, right? So okay, so now we will see what kind of media is used. So as we are dealing here with chitin degrading bacteria, that means the bacteria should have chitin as substrate to be degraded. Okay, so the medium should have chitin present in it. So there are two types of chitins which you can use. The first is colloidal chitin and the another is chitin flakes. Chitin flakes are you will say tedious to handle because the suspension is very viscous and it is very difficult to pipe it out. Okay, and to dilute it if required for particular concentration and etc. So generally colloidal chitin is used, and here I am using, or I have mentioned here about colloidal chitin yeast extract agar. Okay, it is many times also referred as chitin agar simply, right? So this is not just one media composition. If you go on Google and search for some other research articles, you may find there are slight changes in media composition, or you may find some different kind of media composition present. Simply, if you have chitin present in your lab and you don't have some other components, or you are missing out some other components, then you can go with nutrient broth base and you add your colloidal chitin in that and add agar, and you will get your chitin agar ready. Okay, so this actually all depends on what is available in your lab and how much ingredients you have, what all you can use, and you can explore. Okay, so it is not strict that you have to use my media composition which I am specifying here. Okay, you can go with other media which are already used and tested. Okay, so go on internet, google out some good research articles, and if you want to try out at least 2 to 3 different kinds of media, it is also okay. So here the media concentration, or sorry, the media composition is like grams per liter. So Na2HPO4 is 6 grams per liter, K2HPO4 3 grams per liter, NH4Cl 1 gram per liter, NaCl zero point 0.5 grams per liter, then yeast extract 0.05 grams per liter, and agar 15 grams, and colloidal chitin 1% weight by volume. Okay, so now you need to prepare colloidal chitin suspension. So how to go for that? So that is also described here. So if you are new and if you don't know about colloidal chitin, then just follow this procedure. So you take chitin powder, say 40 grams, and you slowly add 600 ml of concentrated HCl. So this actually enhance the dissolution, I can say. Okay, it is very important to use acid, okay, for preparation of colloidal chitin. So you add concentrated HCl and you keep it for 1 hour, 60 minutes, means 1 hour at 30 degrees Celsius with vigorous stirring. Okay, so you need a magnetic stirrer and you keep your flask on that stirrer for at least 1 hour, okay, at 30 degrees Celsius. Now what will happen, the chitin will precipitate as colloidal suspension. Okay, and you make the suspension actually by adding slowly 2 liters of water to that, and the water should be chilled 4 to 10 degrees Celsius. The suspension is collected by filtration with suction as a coarse filter paper. Why you need to filter? Because if there are some lumps or uh, there is some dirt present, then that will be taken out. Okay, so you need to filter by using suction on a coarse filter paper and then washed by suspending it in about 5 liters of distilled water. Okay, so this is also important. You need to repeat washing 3 times until the pH of the suspension is 3.5. And after the above treatment, the loose colloidal chitin is used as substrate by bacteria in medium. Okay, so now you can add this 1% weight by volume in your medium and you make your agar plates. And now those sterile agar plates are one which you can use further for your experiment. So isolation is done by using colloidal chitin yeast extract agar. The culture suspension can be strict or spread plate or pour plate on sterile media plates, and then they are incubated at 37 degrees Celsius for 24 to 48 hours. Okay, so generally you should observe something like this. Okay, so this is, or actually these are some images which I took from some actual research articles, and the credits are given to those authors. Okay, so just to show you how you get your results, I had added these pictures. Okay, so here you can see the plate is labeled as Serratia marcescens. So that is the culture which is used here, and it is showing very good results. Clear hollow of chitin degradation is observed. So this is the chitin plate, and you can see how there is a clear zone of degradation, clear hollow is present. Okay, so this is what you should get, but if these clear zones are not visible, then you can go for staining your plates. Okay, so this is from another research articles where these stains are used to get clear idea about what um, yeah, about the zone of degradation. Okay, so the colonies showing maximum zone of hydrolysis are marked and picked up for making pure culture plates and further testing. Okay, so when you get such kind of results, you just pick that colony for further testing or for making your master plates. Okay, because now you have very good results, and you need to keep your bacterial culture safe. Okay, safe means um, it should be of minimum contamination or no contamination at all, because you need to go further for your testing and identification. Okay, so you need to make master plates. Okay, so that is why you need to pick up your colony and you make your master plates. One more thing I would like to add here is, when you do plate assay method, that means when you use some stains, you should actually go for replica plating, because when you use such kind of stains and your culture gets exposed to these stain solutions, you are actually contaminating your um, culture, and it may happen that your solution, it has, say, like for safranin it has ethanol, so your culture is getting exposed to ethanol. Okay, so now you cannot use that particular bacterial colony for further testing. Correct? So be prepared with replica plates. So if you use one plate for staining purpose, for using stains, then you have same one plate kept to mark which colony gave you good results. Okay, and you will not lose your bacterial culture. So in plate assay method, aqueous solutions of 0.1% Congo red, 0.1% cotton blue and gram sardine are used, and safranin is prepared by dissolving 2.5 grams of safranin in 10 ml of 95% ethanol. Okay, and then adding 100 ml of distilled water to it. So that was in short about the staining solutions. So after incubation, the bacterial plates on chitin agar plates, sorry, bacterial isolates on chitin agar plates, they can be flooded with these stains to get a clear zone. Okay, and allow flooded plates to stand for 20 to 30 minutes with gentle shaking. Okay, so that is how you perform your plate assay method. So plates which are stained with grams iodine, you wash those plates with distilled water thrice, and plates which are flooded with Congo red, cotton blue and safranin, you wash off these plates thrice with 1 molar NaCl solutions for 15 to 20 minutes, and then observe for clear zone formation. Okay, so why this is required? Because it may happen that your solution or your stain is excess in your plate, and there are some depositions on your medium. Okay, so you need to wash for removing those excess stains, correct? So after doing that, you can observe your plates. So for example here, isolate named as NK7. Okay, in all these four plates you can see that is the one which is showing maximum zone of hydrolysis, okay, or maximum zone of chitin degradation. So that is one which you will pick up for your further testing. Another isolate is, I will say, NK11 is also good. It is also showing good zone of hydrolysis compared to the other two. Okay, so this is how you pick up your colony, which shows very good enzymatic results. Okay, so extracellular enzyme activity on chitin agar plate can be measured by. So now you need to calculate the enzyme activity. So you go for this formula. The index of relative enzyme activity, that is RA, is calculated by the total diameter of clear zone and diameter of colony divided by diameter of colony. Okay, the index of relative enzyme activity value of 1 or greater than 1 are classified as cultures having significant enzyme activity. Okay, so the value for RA should be 1 or greater than 1. Okay, so those particular colonies are one which you will pick for your further testing and identification, as those are the one which are having significant enzyme activity. Okay, so that was about plate acid. Now there is another method to quantitate your enzyme, and that is quantitative enzyme method. So for this you need to inoculate your isolates in minimal salt broth containing 1% of colloidal chitin and then incubate at 120 rpm in a rotary shaker at 37 degrees Celsius for 3 days. Okay, so why this? Because as the colloidal chitin is a substrate and it is present in your medium, it will enhance the enzyme secretion. Okay, and after incubation you harvest culture, you centrifuge at 10,000 rpm for 10 minutes at 4 degrees Celsius, and the supernatant is used as crude enzyme. Okay, so as I told you, the colloidal chitin will act as enhancer for enzyme secretion. So now after centrifugation you will use the supernatant that will have the crude enzyme present. Okay, so now you will use this crude enzyme for your enzyme assay. Generally DNSA method is used, or DNS method is used for enzyme assay, but there are some different methods present and recently developed by some researchers. If you go on Google and search, you will find there are 2-3 more methods which are being used for chitinase activity, okay, enzyme activity. So you should know at least the principle and what is the difference between DNS method and that particular method. Try to correlate, try to point out some points and note it somewhere, because in Viva you may get this question. Okay, so the chitinase activity of crude enzyme is assayed by DNS method, which measures the amount of reducing sugar released from colloidal chitin substrate. Now chitin, it actually has an acetyl D-glucosamine residues, okay, and N-acetyl D-glucosamine is actually a derivative of glucose. Okay, so how a reducing sugar? It also has a free aldehyde ketone group, and that is what makes it, or it marks the chitin as it has a reducing sugars. Okay, so when chitin is hydrolyzed and the reducing sugar is released, your DNSA or DNS reagent will mark that. Okay, so that is the basic principle of DNS method. The absorbance of standard and the sample reaction mixture are measured at 540 nanometer. Okay, sometimes you may find that it is 550 nanometers as well, so that is okay, it doesn't make too much of difference. Okay, so the 1 unit of chitinase activity, how will you define that? So 1 unit of chitinase activity is defined as the amount of enzyme which yields 1 micromole of reducing sugar equivalent per minute. Okay, so here the reducing sugar is N-acetyl D-glucosamine. Okay, don't forget. So that is how you define the 1 unit of chitinase activity. So here I had added this flow chart, and I actually liked it. I got it from this particular website, and it is very easy to understand the protocol for DNS method. Okay, so DNS assay is based on the fact that being boiled 3,5-dinitrosalicylic acid, that is DNS, whose color is yellow, it reacts with the reducing saccharide by boiling, and it changes to the brown color. Okay, you can see here the DMS or DNS reagent is yellow in color, and it got converted to the brown. Okay, it should be DNS here, not DMS. So the color change, it indicates that yes, there are reducing sugars present. Okay, so the more the amount of reducing sugar is, the more this coloring reaction proceeds. We can quantitatively evaluate the degree of this coloring reaction by measuring the OD at 550 or 540, as it indirectly shows the amount of reducing sugars. Okay, so that was about DNS method. So these are some other methods which you can read on internet and be prepared for your Viva. Okay, so these are some different assay methods, assay and Shell's method. So these are also used, and you should know about these methods as well. Okay, so lastly, about Viva questions. So here are some which I had listed, and it will give you idea about what kind of Viva questions you may get on this particular topic. So first is, what are types of chitinase enzyme? Then what kind of mechanism chitin degrading bacteria uses for degradation? So that is enzyme mediated, right? Chitinase enzyme is produced. So I will not discuss all the answers, because this will help you to be or it will help you to get prepared for your Viva exam, and that is how you should study. So get a paper and pen, list out all these questions and try to find answers of all these questions. Okay, so mostly all these answers you will find in all my 3 videos of chitin. Okay, and hardly you need to go on Google and search out. Okay, so if you watch my all 3 videos, then I am able to say that you can answer all these questions. Okay, so next is about natural source of chitin, or say you can get a question on chitosan and cellulose, depends. Source of chitin degrading bacteria, or what are the samples which you used for your practicals for isolation. Then what are various applications of chitinase enzyme, then difference between chitin, chitosan and cellulose, then mode of action of endochitinase, mode of action of exochitinase, then give some examples of chitin degrading bacteria, or what particular bacterial isolates you got in your practicals. Okay, all those stuff. Then how is colloidal chitin made, then which stents are used for plate assay method, how extracellular enzyme activity on chitin agar plate is measured, then principle of DNSA method, then media used for bacterial isolate. Okay, then lastly, I thought of this question is, I think it is important, because you should know about different assay methods which are present. Okay, don't just rely on DNS method. Okay, so do you know any other method for chitinase assay? Then yes, I had shared 2 methods, that is Chito assay and Shell's method. So go on internet and check out all about these methods. So then what is the principle of it, then difference between that method and the one which you use during your practicals. Okay, so this is how the examiner builds question on your answer. Okay, so if you say yes, you know about any other method for chitinase assay, then he will ask for the question like, what is the principle of it, and what is the difference between that method and the one which you use in your practical. Okay, so that is how your Viva exam is. Okay, or how your examiner, he starts building up the questions depending on your answers. So be clear about these concepts. Okay, so I hope this video was useful to you all, and thank you for watching. Like, share and subscribe to my channel, and also recommend your friends to subscribe to my channel and like my videos. Till then, thank you and bye bye. Keep supporting. Okay, bye.